Hello, good morning or good afternoon everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Tarek Odalu from Business Review. I will be your host today. It is our pleasure to have Albumedics with us, who will be presenting this webinar, titled Increase Cell Performance with Recombinant Human Albumin. Today's guest speakers are Joaquim Vives, Head of Laboratory for the Development of Cell Therapies, and Phil Morton, Science Director. You'll hear from them in a moment, but first, I'd like to welcome you to our platform. This is On24, and it's fully browser-based. If you do disconnect at any time, just click on the link that you received via email to rejoin the session immediately. All of the icons along the bottom of your screen are the interactive widgets that we have on offer today, so please interact with them all throughout the session. You can ask questions using the Q&A widget or by using the questions box there at the top left-hand corner of your screen. And we'll try to get through as many of the questions as we can once the presentation has concluded. Please use the Help widget if you do experience any technical difficulties or require any more assistance. But for now, please allow me to welcome our first speaker, Phil. Over to you.
Thank you, Tarek, for the introduction, and welcome everyone to this webinar. Today we're going to discuss albumin and its uses within the cell therapy area. I will initially give you an introduction to Albumedics and our albumin products, and then I will hand you over to Joaquim to discuss how they've been using our albumins within their cell systems. The work that will be described later was performed through a collaboration between ourselves, utilizing our expertise in albumin and utilizing the expertise in cellular therapies from the University of Barcelona and Exilia, which is a division of the Blood and Tissue Bank in Barcelona. Albumedics itself is located in Nottingham, UK, where we have both our headquarters and also our GMP manufacturing facility. We are an albumin-based company that produces both recombinant human albumin products and albumin technologies for drug delivery. We have over 30 years' experience in the albumin field, and there are a number of drugs already on the market that use our albumin products. Our longest-serving customer has so far performed over 70 million injections with products containing our recombinant albumins, and so our products are safe to use within the marketplace. So, to begin with, an introduction to albumin. I'm sure you're all aware of some of the challenges that we face when developing formulations for biologics. Appropriate selection of excipients during product development is critical. The excipients are there to stabilize and protect the active pharmaceutical ingredient in the formulation against things like degradation, oxidation, or aggregation during manufacturing, transportation, and storage. While in many cases the use of sugars, amino acids, and detergents are sufficient, there are a number of more challenging biologics where traditional approaches still fail or are suboptimal. The complexity of biotherapeutics has increased with the development of high-concentration antibodies, antibody-drug conjugates, and in particular, and of relevance here, cellular therapies. This has meant there has been an increased requirement for more advanced formulation solutions. And this is borne out with some research that we carried out that showed between 5 and 15% of all formulations require these advanced solutions. This is especially the case with cellular therapies, where cells are delicate and the traditional excipients from the protein field, such as detergents and surfactants, do not work as they can disrupt the cell membrane. Human serum albumin has been included in cell therapy systems, but the variability of albumin batches and from different sources worldwide due to the variation in plasma pools can mean it is suboptimal, and therefore the use of recombinant albumin can bring benefits of the albumin itself while being of a higher quality with improved batch-to-batch consistency, and is of course animal-free. Albumin is a well-characterized and understood protein used in the development of superior biotherapeutics, as well as an excipient. As an excipient, albumin has a wide range of applications in drug formulation, drug delivery, vaccine manufacturing, and as a component of media in vaccine and cell therapy. Albumin's inherent stability makes its handling and storage easier than other proteins, making it suitable for a use as an excipient where other more traditional excipients have failed. We have customers looking at all the fields shown here and have data showing the benefit of use of albumin, but today we will concentrate on the cell therapy area. In terms of that area, we see recombinant can be beneficial across all areas, such as cell expansion, where it can support growth and proliferation, and due to its consistency, can aid in the reproducibility of the cell expansion stage. It can also aid in cell transformation, and where it can support cell viability, as it can within the cryopreservation and formulation areas, where it can aid in improving the product shelf life, and thereby increasing the accessibility of the product, while reducing logistical issues. So at this point, I'll hand you back to Tarek, who will take you through our first poll question.
Thank you very much. Yes, that's right. So it's time for the first poll we'll be running in today's session. So audience members, please go ahead and select the answer relevant to you and then hit submit. So today's polling question is, at what process stage do you see the most challenges or can see the most need for improved methods? So your options are cell expansion, transformation, cryopreservation steps, final formulation, or other. So please go ahead and select one of those and hit submit to vote. For now, I'm just going to hand you right back to Phil to talk a bit more about this question while you vote. Phil?
Yeah, so as mentioned earlier, we do see a use for albumin in all of these areas, and we have data to support in a number of these areas. However, we're really interested to see where people are having the challenges, and then potentially, look at sort of concentrating our research into those particular areas. And certainly if people are interested in collaborations with us, then they're welcome to contact us after the webinar.
Absolutely. I'm just going to go through the question one more time just to give you a couple more seconds here. So at what process stage do you see the most challenges or can see the most need for improved methods? Cell expansion, transformation, cryopreservation steps, final formulation, or other. So please go ahead and select that now. I'll just give you a few more seconds here. Don't forget, hit submit to submit your vote. I think that's all the time we have. Let's move on to the results. Thank you very much for taking part. And here are the results. So it looks like the majority of the audience have gone for cryopreservation steps. So Phil, what do you think about that?
Well, that's certainly interesting and definitely relevant for this webinar, where a lot of the data that Wacom will present later on is around the cryopreservation steps. But as mentioned, if people have got challenges that they see in other areas, then we'd be happy to discuss with you, providing our albumins or collaborations to share with sharing of data. Okay, so in terms of albumin itself, it is the most abundant plasma protein and has the main purpose of maintaining the osmotic pressure of the blood and increasing its colloidal stability. However, it is also used for transporting a wide variety of compounds around the body, as well as protecting the body from toxic compounds. And in fact, HSA can be used to detoxify patients. Albumin is a highly charged molecule, making it very soluble and is very stable over a wide range of pH temperature conditions. It has a long history of safe use, dating back to the mid-1940s, and is non-immunogenic. Consequently, albumin can be used to reduce the shear forces on the cell walls and protect them from the microenvironment. And obviously, with it being human albumin, it is not a foreign compound to the cells. So moving on to our recombinant albumins, here is an example of one of our C of A tests, STS page purity. The results from all of the batches that were tested were above the upper limit of detection, with them all being greater than 99.98% pure, according to this assay. This is showing good batch-to-batch consistency, allied with a high quality, meaning that you can be sure that what you're adding to your cells is the same every time, and that when you optimize your process with these products, you will then get consistency and robustness of your cell systems. Here we have compared a typical sample of our recombinant albumin with that of plasma-derived human albumin. There are some instrument artifacts that are labeled, but the plasma-derived albumin is clearly more heterogeneous than that of the recombinant version. There is a mix of intact albumin with the cysteine 34 free, but the majority is blocked with a cysteine. Whereas the recombinant albumin, the cysteine 34 is pretty well completely free. We've also shown, not shown here, that the consistency of that blocking and also the other components is better with recombinant albumin. It is likely that this heterogeneity in the plasma samples will be different from between different batches and different suppliers around the world, as it is dependent on the donors for the plasma pool and on their diet. Here we've visually compared our two products, Recombinant Prime and Albix, against several other recombinant albumins. We found that the rice-derived albumin tends to be more coloured, while even the other yeast-derived albumins were more pigmented than our products. All the previous data was generated internally, but we also have independent third-party validation of the purity and consistency of our products from Health Canada, who looked at glycosylation levels across several recombinant albumins and also plasma-derived albumin. Not only did our product, which is highlighted here in the red text, have the lowest levels of glycosylation, but they are also the most consistent. When considering the albumin source to use, you normally look at the technical considerations, the cost, and the regulatory acceptance. I've already covered some of the technical benefits of our recombinant albumins in the previous slides. For the other two, they tend to be proportional, with increased regulatory acceptability tending to come at an increased cost. However, as the regulatory environment for these new therapies matures, and the regulators become more familiar with the risks of animal and human-derived raw materials, and the benefits of recombinant sources in terms of better defined and controlled media become more aware, then the extra cost can be offset against the improved compliance and greater regulatory acceptance. So, to summarize, the properties and benefits of albumin: it can act as a nutrient source and protector to the cells, help with transport of critical molecules while also scavenging any toxic or damaging molecules. And all these can help with the performance of your cellular products, while the batch-to-batch consistency can ensure robust manufacture. In terms of the Albumedics products, we produce two forms of recombinant albumin: Recombinant Prime, designed as a biopharmaceutical excipient, and Albix, designed specifically with cells in mind. And it does have a slightly different formulation, which we specifically developed for the cell culture area and stem cells. They are both produced from our proprietary yeast strain and are manufactured to Q7 GMP by Albumedics here in Nottingham. Okay, so I'll now hand you back to Tarek for our second poll question.
Thank you very much. Yes, this is the second poll question we'll be running in today's webinar. So audience members, same as before, please go ahead and select your answer and hit submit. So the question is, do you currently supplement your cell-based products with albumin? So you go yes, albumin, animal albumin, yes, human serum albumin, yes, human recombinant albumin, no, but planning to do it in the next 6 to 12 months, or just no. So go ahead and select whichever one you applies to you, and then hit the submit button. Phil, I'm just going to hand back straight to you there, so you can talk a bit more about this question as well.
Yes, so I mean, as mentioned, we do have a number of customers and a number of collaborators using our albumins, but we are also talking to people and have spoken to people who are using, say, human serum albumin and animal albumins in the early stages of development. And we're thinking, well, are people thinking about moving to recombinant albumins? When in the development process are they starting to think about moving to recombinant albumins? And or are people actually considering recombinant albumins at all? So it'd be very useful to have those, see what people are thinking at the moment.
Absolutely, yes. So the audience, you just have a few more seconds now to vote. Don't forget you need to hit the submit button. So if you've selected your answer, make sure you hit that. One more time the question, do you currently supplement your cell-based products with albumin? I'll just give you a couple more seconds now. Don't forget to hit submit. And 3, 2, 1. I think that's enough time now. So let's go ahead and have a look at the results here. Okay, so look, it's a pretty even split here between yes, animal albumin and yes, human recombinant albumin. So what do you think about this?
So I mean, it's very pleasing that we've got a split and that people are already using human recombinant albumin. Interesting, and we've certainly spoken to a number of people who are using animal albumins as well. And it'll be interesting to see as they progress through the development phase whether or not they'll be thinking about changing from the animal albumins into either human serum or the recombinant albumin. Okay, so now we will go on to some of the actual cell therapy results. And here I'll hand you over to Rakim, who'll go through the benefits that they have seen about using Albix within their products.
Hi, thank you very much. Here connecting from Barcelona, from the Blood and Tissue Bank. As the name says, we are in a blood and tissue bank, so there is a long tradition of working with blood and blood components, but also in the transplantation of hematopoietic progenitors. And most recently, in the last 10 years, we've been developing advanced therapies, basically mesenchymal stromal cell-based products, but also lymphocytes. So this is the result of a collaboration with the Autonomous University of Barcelona, and as you already introduced earlier on, the venture between the two institutions is what we call Excelia. So if we move on to the next slide, here is basically the logistical challenges that we face when we perform developments like this. So from the very beginning, the collection of cells from donors, the starting materials, the cell processing, the transportation, until the moment that we can administrate that product into the patients, because these are living products, and it means that we have to preserve them alive in order to function as a pharmaceutical product. So it's clear that if the viability drops, the cells will not exert their function, and this means that we try to find formulations in which the cells stay alive for a longer time. And if this is the case, then you will see, and this is depicted in the map, that we could have longer times and, of course, the wider areas in which we can use our products. So when we talk about viability, this is the first parameter that we measure, but in fact there are others, because we really want to maintain a given cell number for the treatment, but also that the cells preserve their identity, the purity, and of course the potency. But as you can imagine, working with products that come from individuals, this means that there will be a lot of biological variability. And of course, some of the components that we use are derived from animals, from human serum, and these add some more variability. So the first thing that we did when we had the opportunity to collaborate with Albumedics was to have a look to the regulatory requirements, because currently our products use human serum albumin, so it's isolated from the fractionation of plasma. And what we decide to do is to follow these two guidelines that are shown in the slide, and basically test the recombinant albumins, compare them to the performance of the human serum albumin in terms of viability, the identity, and the purity and the potency of the cells. So here I have the first question. So Tariq, please, if you can take it from here.
Yes, absolutely. So yes, this is the third poll question we'll be running today. So audience members, once again, if you could, please select the answer that's most relevant to you and hit submit. The question is, what cells are you currently using or plan to use in the near future? And the options are embryonic stem cells, mesenchymal stem cells, induced pluripotent stem cells, lymphocytes, other immune cells, none, or other. So please go ahead and select whichever one is most relevant to you and hit submit. Question once again, what cells are you currently using or plan to use in the near future? I'm just going to hand back over to Joachim to talk a bit more about this question while you vote.
Thanks, Tariq. Well, as I said earlier, we work mainly with mesenchymal stromal cells and with lymphocytes. But of course, in this field, there are other cell types of interest, and probably the ones that are listed in this question are the ones that we think that may be the most interesting for the audience today. But it is very interesting to know what are the answers.
Yes, absolutely. So go ahead, audience members, and select your answers and hit the submit button. Just to give you a few more moments now to vote. Question is, what cells are you currently using or plan to use in the near future? So go ahead and select your answer now, just a couple more seconds here. Okay, I think that's given you just enough time now to vote. Thank you very much for taking part in all of today's polls, and let's have a look at the results. Okay, so the majority of you are going for mesenchymal stem cells. What do you think about that?
I think that it makes sense, because it's probably one of the cell types that is widely investigated and also using as a marketed products. But yeah, I'm also I think I find interesting that the embryonic stem cells are also the second one, I think, because there is a lot of potential in the use of ESLs. So let me move on to the next slide, and here we will more in detail into the cryopreservation of the mesenchymal stromal cells. What we did in this study was really to see what conditions are stressing for the cells, and to analyze whether the use of recombinant albumins could add some benefit to these formulations instead of using human serum albumin coming from fractionation of plasma. This is a very summarized picture showing what happens when we freeze down the cells, and there is intracellular ice formation. There is also extracellular ice formation. In the case of the intracellular ice formation, this results in the nucleus function, and in the case of the extracellular ice formation, there are a number of things that happen, like the osmotic stress, cell dehydration, high solute concentration, but also the activation of apoptotic enzymes. In this case, is the caspase family. In any case, all of these conditions lead to the cell death, which is something that we have to avoid in order to preserve the viability of the cells as a medicine. If we move on to the next slide, here there are some characteristics of this specific cell type. They can be derived from virtually any tissue in the body that is vascularized, and here there are only a few examples, from bone marrow particularly, but also from fat, and in our case we also derive the cells from the Wharton's jelly of the umbilical cord. they display similar properties regardless of the source tissue. They have the characteristic that can be expanded clonally, so this is one of the features to become stem cell, and they can also differentiate into different more specific lineages in the mesoderm. In the next slide, we also want to highlight that apart from their multipotentiality, they can also affect local microenvironments. So they have this capacity to respond, for instance, to inflammation or to whatever characteristics are found in the administration site. And the molecules that are secreted by the MSCs can be bioactive and, of course, can work as a drug. So going back again to the design of the experiment, what we did in the very first one was to expand the cells. We were using clinical grade mesenchymal cells. We expanded until getting sufficient numbers. And we performed two types of study. One was using a fresh product, and we tested 2% weight volume Albix compared to human serum albumin. And in the other case, we freeze the cells, also using two different amounts of Albix or human serum albumin. Then we thaw the cells and studied the stability. When we did this, we found that the stability at 2 to 8 degrees for the cells that were not cryopreserved was quite similar. So as you see in this image in the top panel, the results using Albix 2%, we could see the number of dead cells on the top, viable cells on the bottom, and it was increasing a long time. But that was quite similar to the results that we got using the human serum albumin. The same results are also shown on the right of the slide with bars. So, okay, this was good because it meant that we could use Albix as a substitute of human serum albumin. And if we take into account that the consistency of the different batch-to-batch productions of Albix, this was a very positive point, but it was not adding any extra benefit with respect to what we were doing until now. So then we decided to study what happened when we thaw the cells that were cryopreserved, either with Albix or with human serum albumin at the different concentrations. And here it was surprising because we saw that Albix was really beneficial for the cells. And it was at the concentration of 2%. Adding 5%, it didn't improve the results. Actually, the results were slightly worse, especially for the use of human serum albumin. In the next slide, these results are shown in numbers. And I think that here is much clearer the result in the consistency of the use of Albix along time, giving viabilities up to 92% at 48 hours. So once we found these results, we decided to dig in a little bit more and try to understand what was going on. So we focused really on the cryopreservation protocol. We discarded to work with the fresh products, and we just as a summary of what we do, we usually cryopreserve the cells, 10% DMSO. We always add 2% albumin, and then we use an isotonic electrolyte solution. It's called plasma light. And under these conditions, after thawing, we have a shelf life of 26 hours. So this is the target that we wanted to improve. If we move on to the next slide, this is exactly what we did. We cryopreserve MSCs using Albix or using human serum albumin, again at 2%. We freeze them down, and then when thawing, we use Albix or human serum albumin in each of the different situations. And then we took the samples, and this time we performed a very comprehensive characterization. It was not only the viability, but we also analyzed the identity by a phenotype evaluation, the potency. We really were interested to know whether these cells retain their capacity to differentiate. And then, of course, the viability again. If we start with the identity, here in these images, we show results at day 0, time 0, and also at 72 hours later. And the results were very, very similar when we use Albix and human serum albumin. So despite of a decrease in the viability that happens a long time, the identity was preserved in both conditions. If we move on to the next slide, here there are some of the results of the differentiation assay. And what we demonstrated with this study was that the osteogenic, the dipogenic, and the chondrogenic potential of the MSCs was preserved with the presence of Albix. These results, and we couldn't show here because the panel was huge, it was also similar with the human serum albumin. The difference is, and this is something I'm showing in this slide, is that the variability was much, much, much lower. So this means that despite the potential and the identity was retained in both conditions, only with the use of Albix we could preserve a very, very high numbers. So it was surprising to see that the stability of the cells was extended up to 72 hours. Here you can see also the standard deviation, because in this case what we did is also, in order to comply with a proper validation with the GMP, we used 3 different GMP grade cell lines. As a conclusion of this part of the talk, Albix preserves cell viability after thawing, and this was significantly better compared to human serum albumin. But finding these results, it was again surprising to us, because what was the mechanism? At the end of the day, we were using protein instead of using human serum albumin coming from the fractionation of plasma. We were using a recombinant protein. So we wanted to understand mechanistically what was going on. So we performed some apoptotic studies, and we analyzed a number of cells that were in an early apoptotic stage or in a late apoptotic stage. And here is when we saw differences. So in terms of apoptosis, we didn't see the difference. The differences came in the percentage of cells that were early or late. It looks like Albix may prevent the cells to enter into late apoptosis. So there is an increase of cells in early apoptotic stage. So as a conclusion of this study, the first important thing is that the use of Albix, it really met our criteria, and so we could use it also for the stability of a fresh product, because no significant differences were found between Albix and human serum albumin. But after cryopreservation, after thawing the cells, we could extend also the stability of the product. So this means that logistically, this is much favorable to us. And here we have the summary again of the results that I just said. So now, if we move on to another of the studies that we performed, because we said, okay, look, this is working with MSCs, and it looks like it's working in those parts of the bioprocess where there is something that is very stressing for the cells, and in this case, it's the cryopreservation. So at that time, we thought, okay, we have another product that is based on the lymphocytes, and along the bioprocess, there are also different steps that are, or we think that they are, very stressing for the cells. So, okay, we use currently the human serum albumin, and this is for the generation of virus-specific lymphocyte, and is a medicine that we are currently under development and a clinical trial. The production process, as I said, involves steps that are highly stressing for the cells, and this means, for instance, the stimulation of the lymphocytes, the isolation of interferon gamma secreting cells, but also the conditioning of the final product. So we thought that here we could apply Albix and see whether their protection could be much higher than the one that human serum albumin is doing. This is a very easy schematic showing how we do it, how we produce the cells. And I'm going to give a very, very short summary of what this means. Basically, you need to think in a patient that has been receiving a transplant. It could be a solid organ, or it could be hematopoietic progenitor cells. In these conditions, the patient is immunosuppressed. And here there is an opportunity for virus, like cytomegalovirus, Epstein-Barr, adenovirus, either to reactivate, or there is a de novo infection. If that is the case, of course, clinicians have a lot of different drugs that can use in order to manage the titer of viruses in the body, but in many cases this doesn't work. So we are following a protocol that actually is used in many other places, in which what we do is try to isolate memory lymphocytes from a donor, and then to expand them in the lab and reinfuse in the patient. The problem we have is that the shelf life of this product is only 6 hours. So we really were interested in trying to extend the lifespan of this product. Here there is a very simple summary of the experimental design. Basically what we do is that we take some peripheral blood from the donor. Then we perform a gradient in order to isolate mononuclear cells. We perform some washings. Then there is a counting. After this, there's a stimulation of the lymphocytes. In the case of the actual drug, what we do is that we perform virus-specific stimulation. We stimulate either for memory lymphocytes for cytomegalovirus or adenovirus, as said earlier on. In this specific study, what we did was unspecific stimulation. So we use CD3, CD28. And then the next step would be passing the cells through a column, in this case is a Miltenyi column, in which we would retain only the cells that have been activated. And we know that these cells are activated because they secrete interferon gamma. So in order to simulate the conditions that we currently use with the human serum albumin, what we did was to use exactly the same amounts, but in this time was with Albix. Here, the difference compared to MSCs is that instead of using 2%, we commonly use 0.5%. So we perform exactly the same conditioning and then compare using these assays that are shown on the right of the slide. Cell counting, cell variability, apoptosis, and polyclonal activity. And we did it, of course, at different times, taking into account that the shelf life is very short. And here we got the first results pre-column. So this means that we stimulated the cells and then analyzed what happened for the whole population, and this is what we can see in the left panel. There is no difference really between the use of human serum albumin or Albix, and is also very similar results for the use of the albumin from Albix or human serum albumin in CD8 positive cells. Then we analyze CD4 positive cells and also the CD4 positive cells secreting interferon gamma. And again, the results were quite similar. So as we said previously for the MSCs with a fresh product, there is no difference. So in fact, this is good because it means that we can maybe at some point switch from human serum albumin to a product that is more reliable and consistent from batch to batch. But we couldn't see any improvement. And then we analyzed apoptosis because, as we shown earlier, the mechanism of action was based on that. But here we couldn't see any difference. Probably as a result that previously we didn't see any difference for the CD4 and the CD8 positive cells. The next step was to pass the cells through the column and see whether this step was more stressing, and maybe there was a difference in the viability of the cells. But again, the results were not different. So we have to say here that Albix performed the same as human serum albumin. With these results, we concluded that recombinant albumin provides the equal viability as the human serum albumin, but adding this consistency and security that Phil showed us earlier on. There are, of course, points that maybe we have to consider when we end in this conclusion. One is that we miniaturize the setup. So, of course, what we didn't do was to use the clinical scale in the study design, and these conditions may not be as stressing as the clinical scale. Also, I wanted to say that the stimulation was on a specific. In this case, we stimulated all the lymphocytes by using a CD3, CD28. So maybe virus-specific could be more stressing because the cells need to duplicate much more in order to get sufficient numbers for the infusion. So these are things that we would like to study in the future, but so far, this is the conclusion that we have today. So as an overall takeaways, we have these 3 points. Basically, that Albix is a highly pure and consistent recombinant product which offers security and reliability, that it exerts a protective effect on cells under situation of stress and results in this higher viability and longer stability times. So these allow us to solve some of the logistical problems and having more time from the release of the product until the administration. And with this slide, I complete my talk, and I hand it to Tariq.
Yes, thank you very much. Audience members, we're about to enter our live Q&A session now. So if you do have questions, all you need to do is type that question into the box there at the top left-hand corner of the screen and hit submit. We're going to try to get to as many of the questions as we can. So we've had a few coming in so far. Let's start with one of these. So this person has said, what are the biggest benefits of the increased viability of the MSCs after cryopreservation using Albix? Who wants to take that?
I think Joachim, is that something for yourself?
Yes, sure. So the biggest benefit of the increased viability for us, well, this is the most powerful result, because the problem we have now is that the shelf life of these products is very, very short. Basically, we're talking about 26 hours the most. We have other products like the Lymphocyte that is 6 hours. We have fresh products that is 18 hours. So the more that we can extend the shelf life, the more flexibility we have in order to coordinate with the team in the surgery room, also with the patients. You have to think that when we perform clinical trials, sometimes the patients are not from the same city. They have to move from other places. It could also help us to coordinate multi-center studies, because then this means that we can ship the cells to longer distances. So to me, this is probably the biggest benefit.
Okay, great. Thank you very much for that answer. We're going to move on to another question. Audience members, please do keep the questions coming in. Thank you very much. Next question is, why have you used your Albix product throughout the studies and not Recombinant Prime? Who wants to take that one?
I mean, I can take that one initially. So as mentioned earlier, we have both Albix and Recombinant Prime. Albix we have specifically designed for the stem cell market, and we went through a number of tests checking its functionality within stem cell systems. So we primarily see Albix as the product for stem cell systems. However, we do have customers using both Albix and Recombinant Prime, and there are some differences. So it's quite often beneficial for the customers to try out both products and see which is best for their systems and at different places. So I mean, one product might be beneficial within the expansion part, whereas another product may be beneficial, say, in the differentiation part of their process.
Thank you very much. Let's move on to another question now. So this person has sent in this question. What levels of cell loss do you normally see in the clinical production process of the T lymphocytes? Who wants to take that one? Okay,
yeah, I take that one. Well, our specifications for these products is that the viability should be higher than the 70%. And what we found is that after 6 hours, the viability drops from that number. So this means that the total number, of course, is reduced. And this, again, as I also said in the previous question, is a problem. But because what we want to do is to preserve the number and also the viability of the cells, so that they can exert their function.
All right, thank you very much. Audience members, please do keep the questions coming in. So don't forget, you need to do that by typing your question into the box there at the top left of the screen and hit submit. Next question we have here, this person has said, do you have data on your products for expansion of cells as well?
Yes, so I can take that. So yes, we have got data for the cell expansion stage. We've used in combination with other collaborators, Albix with 2 different cell lines, again with mesenchymal stem cells, but also with human embryonic stem cells. And here we've compared it to other recombinant albumins and also human albumin. And we have demonstrated that our recombinant albumin gives better results, certainly, than the other recombinant albumins that were tested alongside, and gives at least equivalent, if not better, results than the HSA controls. That data is within a slide deck, which I believe is available to download, but also you're welcome to contact ourselves after the webinar if you would like that data.
Okay, great. Thank you very much. Another question we've received: why do you add Albix to the MSCs both during cryopreservation and afterwards during the thawing as well? Do you see a big difference if not added during thawing? Who wants to take that one?
I'll take that one, Tarek. Well, this is a very interesting question, because what we analyze here is the use of Albix both at the cryopreservation step and also in the conditioning solution that we use after thawing. But I didn't have time to show more results in which what we did was to switch conditions. Let's say first we cryopreserve the cells with the human serum albumin, and then we thaw it using Albix, and just the opposite, the other way around. We use Albix for the cryopreservation and then human serum albumin for the conditioning, and then following up the stability. And it's very interesting because our conclusions are that the longer the contact of the cells with Albix, the higher the viability. So all these results lead to say that if Albix is used in the cryopreservation and then again in the final formulation, the viabilities are the highest.
Thanks a lot for the answer there. And we're going to move on to another question now. Audience members, please do keep the great questions coming in. Thank you very much. We're going to try to get to a couple more here before we wrap things up today. This question is: what kind of regulatory documentation do you have on your products?
Yeah, so I can take that. So for Recombinant Prime, we have a full biologics master file that we have registered with certain regulatory agencies. Because it's used as an excipient, not all regulatory authorities recognize that, but where possible we have registered that with the authorities. That has both preclinical and also we have some phase 1 clinical data within that for Recombinant Prime. For Albix, we've got a file at the moment that we have ourselves. We are in the process of developing a master file, and as part of that, we're currently doing some preclinical tox studies. And once we have all of that data together, we are looking to sort of generate a master file for that as well.
Okay, thanks a lot. We're going to move into the next question here. So this person has said, going back to the impurities in the various sources of albumins, what are common impurities, i.e. heavy metals, lipids, or associated proteins? And is this information available for testing per lot of Albix? Who wants to come in on that one?
Yeah, so I can discuss that one. So for our recombinant albumins, the primary impurities are things like host cell proteins. We produce our products within our Saccharomyces system, so they're yeast host cell proteins. We do also measure and have measured in the past during the process qualification the full range of metals. We don't do a full metal test for every single lot, but we have got process qualification data around heavy metals and things like that. Lipids, they will be stripped out from the process during the purification process, and so it's really the host cell proteins that are the primary contaminants and impurities. But even there, those levels are extremely low. They are on a lot-to-lot basis, and they are well within what's generally accepted for clinical grade materials.
Okay, thank you very much. The next question we have here, so this person has asked, do you offer recombinant HSA, that's human serum albumin, in powder?
So yes, so I can take that as well. So at the moment, no, both of our products, Recombinant Prime and Albix, we offer in liquid formulations at either 20%, so 200 megamil for Prime, or 10%, which is 100 megamil for Albix. They are in either sort of 50 ml vials, or for Albix, we can also provide in bioprocess containers as well for larger volumes.
Okay, great. And the next question is, this person says, I would like to know more about immunogenicity of Albix if used for transfusion. Can anyone come in on that one?
Yep, okay. I can answer that as well. As mentioned, we're in the process of performing tox studies with Albix at the moment, and that will go into a regulatory package. The intention is not per se to do clinical trials with Albix. We have done phase 1 clinical trials with Recombinant Prime and shown it to be non-immunogenic. Albix is from a very similar process with a slightly different formulation, so we would be expecting that to be also non-immunogenic. It is human albumin itself, so the actual product itself will not be immunogenic, and we are testing the impurities within it within a tox study.
Okay, thank you very much for that answer, and thank you to everybody who submitted a question today. Unfortunately, we are all out of time. I'm very sorry if we couldn't get to your question, but that is the end of today's session. So that just leaves me to thank our speakers for what was a great presentation, I'm sure you'll all agree, and to thank Albumedics for sponsoring the session. To the attendees, you will receive an email tomorrow that will tell you how you can access the on-demand version of the webinar. You can get that on our website, which is www.business-review-webinars.com. And we do look forward to sharing further webinars with you in the future, so if you'd like to keep an eye out on that website, you can also follow us on Twitter at BRWebinars for daily updates, and you can join our LinkedIn group as well, Business Review Webinars, to stay up to date. Thank you all once again, and I hope you all have a lovely day.